So reason I have the microphone with me and this is hooked up with the microphone. I think we wanted to have this session recorded, especially the presentation part of the defense, and I'll make this available for our faculty and colleagues who were not able to be here physically. So with that, I would like to thank you being here and joining the oral defense as we are getting closer to the end of the semester. I know everyone is busy and with your end of semester activities, but thank you so much for being here. And I would like to also thank our committee members, Dr. Ling Ann from Agriculture and Biosystems Engineering Department, as well as Dr. Joel Coelho for willing to serve on Alex's committee. I would like to also say hello and welcome to Alex's mother. She's also here with us to celebrate the achievements of his son. And before I kind of give the microphone to Alex, I would like to say a couple of things about this program, because this is slightly different than our traditional master's and PhD programs in the department. Alex, the degree program that Alex went through is called a Professional Science Master's Program, which is a program under Applied Biosciences of Graduate Interdisciplinary Programs of University of Arizona. And under Applied Biosciences, we have four tracks. These are, I'm going to cheat here, medicinal and diagnostic laboratories, molecular and cellular biology, and industrial microbial biotechnology, and controlled environment agriculture is one of them. And we are fairly new program, degree program under this biosciences in GIDP. And currently we have four students, and Alex and Sean McBride, and we have Don Kuhn, and we have another student, Kevin McCurry, currently going through this program. And this program is pretty much for students, crafted for students who would like to have a graduate level education, really interested in integrating and engineering and science related to controlled environment agriculture, and wanting to work in that industry. They have the opportunity also to develop a background in business and management aspects of this emphasis area. And they're pretty much on the driver's seat. So it's really flexible program. They decide on what courses they would like to take and how they would like to grow their kind of skill sets and background in this area. And it really puts emphasis on the internship. And internship is really critical, and working either in a research lab here on the campus or maybe with an industrial company out there, commercial company out there. And the main goal is basically to contribute to the activities or the needs of that lab or the company, wherever their needs might be. And during the process, it is expected that the students are able to integrate their learning into that hands-on experiences. And during the defense, they basically defend that internship activity. So Alex went through this route, and he kind of stepped up for the challenge. Usually we see students going for only one internship, but Alex actually went through 3 internship activities, 2 of them here on campus and 1 in Chiba University in Japan. So he's going to briefly talk about that. So that's a brief information about the program. And I would like to say a couple of things about Alex. Some of you may not know some of the things that Alex has in his CV, and I know most of you know him fairly well. So Alex actually was born and raised here in Superior, Arizona, where this is where their family actually live and moved to. And his father was the director of Boris Thompson Arboretum there, a beautiful arboretum in Superior. And Alex graduated from Arizona State University Polytechnic campus with a Bachelor of Science degree in Applied Biological Sciences with emphasis on sustainable horticulture, minoring in sustainability as well as computer sciences. And I also learned that he spent time in Australia through a sustainable agriculture and global health program, and he also went to Peru to help building a small greenhouse for an orphanage, I believe. And then he was interested in vertical farming and controlled environment agriculture. That's when he joined the program back in 2015. And for the past 2 years, I had the pleasure of working with him and acting as his supervisor. So we're going to give a chance for him to tell us what he did during his degree program. And this is going to be the presentation for you for about 40 minutes. Then we will allow questions from the audience. Then we will kindly ask for audience to leave, so the committee can spend a little bit more time kind of hassling with Alex, challenging him about what he did. Okay, again, thank you very much. Alex, it's your turn.
Thank you, Dr. Kachira. And thank you all for coming to my presentation today. I'm really excited to share my work with you that I've done from May 2016 up through August of 2017, focusing on optimizing crop production and resource use efficiency in vertical farm systems. So I do have a lot of material to cover today, so I'm going to try and get through it and stay on time. I'm going to start with a brief introduction, just kind of going over some of the motivation and challenges in North America and worldwide with food production and logistics. I'll go over the current state and challenges in the vertical farm industry and the need for vertical farm research. And then I'll give an overview of the objectives of my internship curriculum and get into the details of each portion of the internship that I did. And then I'll do a little bit about other achievements while I was here at the university and, of course, acknowledgements. So diving right in, and for some reason I can't use the clicker. So the United Nations projects that there will be 6 billion people living in urban areas, living in cities by the year 2050. A total of 10 billion people are projected to live on the planet by that year. So a lot of people will be in the cities and they will all have a demand for fresh food. I cannot get this clicker. So there's a need for complementary integrated systems in order to produce food sustainably for now and the future. Greenhouse agriculture is 10 times more resource efficient than open field agriculture, and indoor agriculture is 10 times more resource efficient than greenhouse agriculture. So controlled environment agriculture systems such as greenhouse and indoor, when combined with open field agriculture, can help address these food imbalances and strengthen the food-water energy nexus that we're hoping to create in the future. So a little bit about food miles. The weighted average source distance for romaine lettuce arriving by truck to Chicago is just over 2,000 miles. That's a study from Iowa State in 2001. Coincidentally, Yuma, Arizona is about 2,000 miles from Chicago, Illinois. And if I just jump back a bit, I did have just representations of the different forms of crop production. Open field lettuce in Yuma, an example of greenhouse production of floating raft lettuce, and shipping container-based indoor farm there, the Growtainer. So again, this could perhaps be done better by positioning greenhouses and indoor farms closer to these population centers rather than growing it all in Yuma, even though that's great. So just a quick overview on vertical farming. Vertical farming is a form of producing food and medicine crops in vertically stacked layers using hydroponic systems, usually also using artificial lighting. So that can be a shipping container, a warehouse, or even a multi-story, multi-level building employing this technology. Again, kind of referencing back, this uses about 1% of the resources, particularly water, compared to traditional open field agriculture. Another name for vertical farming is plant factory with artificial lighting, or PFAL. That's the term that's used in Asia. Japan is acknowledged to be the birthplace of this industry. The first PFAL actually dates back to 1983 in Shizuoka Prefecture in Japan, and now they're up to 200 plant factories as of 2015. So this actually is over a 30-year-old industry already. And at this moment, there are about 90 to 100 vertical farm and container farms in the United States as well. And we just have here an image. It's an example of the Mirai facility in Japan, where I actually did part of my second internship. So a little bit about the productivity, revenue productivity of these different types of farms. The most revenue productive farm, this is a study that was conducted by Agrilist. It's a report that they put out called State of Indoor Farming in collaboration with Cornell University and Urban Ag News. They surveyed over 150 indoor growers to get some of this information, and so I have a few slides on that. Indoor cannabis is currently the most revenue productive form of farming, followed by indoor leafy greens and herbs. So you can see there's quite a noticeable difference. This is on a per acre basis in how revenue productive these farms are and can be. And another way of looking at that is crops grown by facility. So if we focus on the vertical farm and container farms, it's a little hard to read, but those two columns on the right are leafy greens, herbs, and microgreens. So that's the majority of the crops that are being produced in these types of facilities because they're the most revenue productive. So some top rank operational challenges that Agrilist returned from their survey of 150 indoor farmers. The very top challenge is actually keeping their operating costs down. So operating costs are high in this type of facility, and that their number one concern is keeping those costs down. I'd also like to bring your attention to managing farm labor and predictability of plant performance. Those are also concerns that are quite prevalent in this industry. So if we can improve those things, it would be very beneficial. There is absolutely a need to reduce the labor input in a vertical farm, as well as a need for increased automation, monitoring, and control to reduce data analysis time, and of course improve our crop yield and quality. My image on the right is on data analysis time. The x-axis is farm size, and so you can see these very small farms are in fact using a large percentage of their labor time to analyze this data. So there would be a big improvement if we can work on that. And just a little bit of figures about the what labor looks like in an indoor farm on the left. So another motivation that we have is what's called tip burn, which is basically where the sort of center core of the lettuce head has necrosis and is damaged. It affects crop quality and yield. This is actually a calcium deficiency, which is caused by inadequate transpiration due to a lack of airflow to the canopy. So the core is not able to transpire and bring that calcium to the tip. So it's not because there isn't calcium in the water or in the nutrient solution. So again, this reduces resource use efficiency, labor use efficiency due to the extra time needed to deal with this, and also the resources that are kind of thrown away in the process of mitigating this. So there's absolutely a need to develop an improved airflow distribution system for vertical farm systems. So there's actually no commercial scale research facility that existed in the country at the time that the UAG farm facility was launched, and that's still true to this day. We're not aware of any other commercial scale research facility in the country for vertical farming. So our goal with launching the UAG farm facility that I'm going to discuss in detail was to actually develop a research, education, and outreach program at a commercial scale using collaboration and sponsorship from the industry to address some of these challenges in vertical farming. So the Controlled Environment Agriculture Center, Dr. Kachira's lab, the Advanced Sensing and Climate Control lab in the Agriculture Biosystems Engineering Department, partnered with industry sponsors Illumitech, Civic Farms, Hort Americas, and Indoor Harvest, and with some funding from the Water, Environmental, and Energy Solutions Program to remodel an existing building that we had here at the SEAC and turn it into a research grade facility. So expected outcomes from this are to conduct engineering and science-based research at commercial scale, creating a skilled workforce for this new industry, and performing outreach on and off campus to inspire students and stakeholders about this industry. So just a quick overview of the sort of big picture of my internship curriculum. Again, like Dr. Kachira mentioned, there's a strong emphasis on hands-on industry sort of research experience 360 hours of internship are required for our degree. I have an interest in urban farming solutions and technologies improving the efficiency of these farms. So the first portion of the internship was spent here at the Controlled Environment Center in Dr. Kachira's lab, developing a data acquisition platform for monitoring and control of the UAG farm. The second portion was done at Chiba University through the Japan Plant Factory Association, where I worked in 2 different commercial scale vertical farms, gaining hands-on experience with daily practices and logistics there. And then I finally came back and carried out an experiment here in the UAG farm, evaluating the effects of light intensity and fertilizer type on lettuce and basil crops, and also gained management experience working with graduate and undergraduate students as part of the team. So getting into the first part of my internship here, just a quick bit of information about the UAG farm itself. It's a 770 square foot facility with 2 chambers separated by a wall and a sliding door, individually climate controlled by a heat pump heating and cooling system. There are 2 hydroponic systems we call modules in each chamber, so a total of 4 modules. Each module contains 3 deep flow technique or floating raft hydroponic growing beds. It's sort of a hybrid system between nutrient film and deep flow. There are 144 planting sites per growing bed, per 4 by 8 foot bed, so we end up with 4.5 plants per square feet density. Each bed is illuminated by 8 LED grow lights generously donated by Illumitex. They're roughly mostly red light with a small amount of blue and green as well. There's, of course, a water pump on each module, and each chamber has 1 air pump shared between the 2 to provide proper conditions and circulation. And so this, you can see, provides an opportunity to perform multi-factor research experiments at a commercial scale while training our workforce, while training our students. So, sorry, I'm having a little trouble with the clicker here. Our objective for the first internship was to develop, install, and implement a sophisticated environmental and resource monitoring, control, and automation platform to enable research in this facility while improving our crop yield and quality on a continual basis. So specific requirements were collect the key environmental variables and resource use variables, use that sensor feedback to control hardware in order to manipulate our variables, totalize any resource consumption so we can do economic analysis, create a graphical user interface to empower the facility operator so they don't need to know the ins and outs of the program coding and so on. They're able to use the software to do their work, and also develop some additional hardware components that were necessary for the facility. So I have here a small image of the computer control room in the UAG farm. We have a 3-monitor computer that we use for our sort of command center, and a nice window to peek into one of the chambers there. And also the gray box there is our enclosure containing our data acquisition system. So a little bit more detail about our key variables and the sensor feedback. My flow chart here, it's a little hard to read, but essentially what's going on is we have our variables from each chamber, from the aerial environment and the root zone, temperature, relative humidity, carbon dioxide, light intensity, EC, pH, dissolved oxygen of the water. All these variables are being collected every 5 seconds and sent to the data logger, as well as some pulse counting from our environmental variables and any manual controls sent from the graphic user interface. All that is sent to the data logger, and then it runs its program and determines whether or not to actuate any relays in order to control any of these variables based on the program code that I developed. Just a few examples of some of our sensors you can see there, as well as a custom aspirator that I built with the help of Dr. Jeff Feja, and our custom fertigator unit as well in the corner there, containing 2 peristaltic pumps and 4 solenoid valves that enables us to automatically inject concentrated nutrient solution or a nitric acid solution to correct our EC and pH. So this is a distributed signal monitoring, processing, and control system that I developed. There is a graphical user interface, as I mentioned. If you imagine 3 screens like I showed you, the first 2 are each dedicated for each individual chamber, and the third one is a resource accounting view. So the first 2 chamber screens have several tabs, different ways you can view the information. There's an instantaneous data view that enables, at a glance, understanding of all the current state of all of our environmental variables, as well as a historical view that can be switched to see a sort of 24-hour trend to ensure that we're keeping with our set points as we define them. And then there's also a sort of debug and manual controls page that allows some tinkering with the different set points and injection times, as well as manually controlling some of these relays. And then the third monitor is dedicated to resource accounting, so we can at a glance see the consumption of power, water, and carbon dioxide, and see how the 2 rooms compare to each other. So that's just a brief overview of the first portion of my internship. So we ended up with a fully functional data acquisition system that does sensor feedback control with a graphical user interface. So next, I traveled to Japan. Dr. Cherry Kubota helped arrange an internship through Chiba University and the Japan Plant Factory Association. The objective of that internship was to gain experience with production and logistics in a real-world commercial plant factory. I spent 5 weeks working at Mirai, which is a 10,000 heads per day commercial facility, and 2 weeks working at Ray's Farm, which is like a smaller scale, about half the size of Mirai, research-focused facility located on campus at the Chiba University Agriculture Campus. So on the left we have Mirai's lettuce room, and on the right is Ray's LED demonstration testbed. So first about Mirai. It's located in Kashiwa, Chiba, Japan, just biking distance away from the campus. It was built 3 years ago. They actually moved off campus and started this new facility. The building on the right is their production facility in my top photo, and on the left is their office building. So they employ 35 part-time workers. Roughly 20 workers are working on any given day. They only have 3 full-time supervisors and several office staff, of course. So at the time of my internship last year, the facility was still primarily using fluorescent lamps, but they were planning to switch to LED in late 2016 for their lettuce room. They do have 2 identical rooms. A room is for lettuce, B room is for basil. They're producing 3 different varieties of lettuce, romaine, a green leaf, and a frill lettuce, which is quite tasty, and 2 varieties of basil as well. And they do have slightly different harvest cycles, an extra week for the basil because it does take a little bit longer. Each room contains 11 of these modules. Each one is 1 meter wide by 10 meters long, and there are 10 tiers to each one as well that are spaced about 40 centimeters apart. So it's quite densely planted, and they sell their product to distributors and direct to grocers on an order basis. So when orders come in, then they produce for their buyer. And they do have a goal to expand their production through franchising the Mirai name, creating more of these facilities around Japan. So I performed a lot of different duties as a part-time factory worker. They were kind enough to rotate me through basically every position in the facility to get more broad range of experience. So seeding is done into these urethane sponges. That's a small sponge cell that can be torn apart. They have a nice seeder that enables you to do a full tray in about 1 or 2 minutes, which is really nice. Transplanting is done into 1 by 2 foot small foam rafts. So you can just you dip the cube into some water and do a kind of dip and squeeze method to direct all the roots into the hole. So they've got it all very precisely figured out. They've been doing it for a while. Harvesting mature plants as well is a big part of every morning. They bring down panel after panel. You remove the cotyledons, any damaged leaves, so you're left with like a perfect quality head of lettuce or a bunch of basil. They're loaded in a very particular way into these stackable bins and roll off to the package room where they're harvested. Lettuce is automatically packaged, but the basil is manually done by weighing it and sealing it using a heat sealer that I got experience with using. So all those tasks happen every day. They're on a continuous rolling cycle, and they do have some machinery for cleaning panels, packaging the lettuce, and of course their lifter to grab those panels down from the higher levels. So Raise was the last 2 weeks of my internship, which is located on campus at the Chiba University Agriculture campus. They have a facility called P Square it's a collaborative effort between industry and university. So I was there 2 weeks. It is research oriented, so they're doing research on testing the different manufacturers of LED lights, looking at quality and even taste, how taste is affected by these different light qualities, and as well as tip burn mitigation. They're looking at employing different strategies in order to reduce the amount of tip burn that they get. Their system is basically identical to Mirai's, and similar with the production workflow as well. The only difference is they have denser spacing to their racks, so they do see a little bit more tip burn. So that's an additional labor component there, is the tip burn removal during harvest, as well as cleaning the panels just using a power washer instead of a large piece of machinery. So in the Raise farm, it is research oriented, and they are collaborating with a company called PlantX, which is an engineering controls company that has a product called Saibai X. Saibai means cultivation. So Saibai is a monitoring and control platform employing software and hardware in order to just provide decision-making support to the operator. You can see here they have a sort of demonstration window here where you can see they have a 9-monitor display viewing webcam and all sorts of different variables there. It's a very powerful program referencing 500 papers by Toyoki Kozai, who's considered to be the father of the plant factory industry, and it uses 250 real equations from Kozai's papers, not like correlated things or anything like that, the real equations. So it's a pretty impressive product, and they are planning to expand it to actually include the entire supply chain. Everything from labor to materials is ultimately going to be included in this Saibai X. So it's an interesting industry collaboration there where they're generating this research, testing their platform, and so on. So I did want to speak a bit about the sterility or clean room practices in the plant factory. At Mirai, I counted it, I have to get through 8 doors to get in the facility. So I thought it'd be worth bringing some attention to just how strict their clean room practices are and how effective it is. So the first room is simply for taking off your shoes and storing those. The second room is where you hang your jacket, use the restroom. Then it splits off into men and women. You actually strip down to your undergarments, put everything in a locker, go through another door which has water showers, which are in case of emergency. They weren't used on a daily basis. Then you put on work clothes provided by the factory, a clean suit, mask, hairnet, really covering up well. You can see that's me in proper attire. And of course, work shoes in the following room, followed by an air shower, which you can see on the right there, just in case something's on you, even though you rollered your jumpsuit on the way in. So very proper clean room practices. And on the way out, all of the clothes go in a washing machine where they're cleaned and ready for the following day. Raise was a little bit less strict. You could just wear your own clothes and put the jumpsuit on over it. Some of the rooms were combined. But neither facility had any insects at all, which is incredible. They didn't even have sticky tape up to catch them because there were none. So clearly this is effective. And there was virtually no mold present, and extremely small presence of mold, which they have a procedure to get rid of to ensure it doesn't spread. So very high clean room practices there. I did sort of have some observations from spending my time there. Definitely some room for improvement, which is a good sign. There is a high energy consumption due to the uneven air temperature distribution. If you noticed in my 3D image of the temperature, there are some kind of hot spots in the center. That's going to be where you get tip burn, and you're going to have to cool even harder to try and get this air to mix. Definitely high labor cost and waste due to the quality standards. I spent a lot of my time throwing away what seemed to me like perfectly good food, just in order to keep it to the quality standards that they needed. This could perhaps be recycled into either a heat source, energy source, or fertilizer source. The major pest is actually algae. These systems are fantastic for growing algae. It reduces the crop quality and harvest weight because of algae on the leaves that have to be thrown out. Labor cost is increased from having to spend time every day draining algae from these panels and removing it manually, which of course makes you throw fertilizer away in the process and the water itself. Some control strategies I discussed with Dr. Kozai would include somehow basically sterilizing the nutrient solution through either hydrogen peroxide, ultraviolet sterilization component, or even like a hot water boiling component are all ways that this could be dealt with. But ultimately a redesign of the hydroponic system itself so that it doesn't grow algae in the first place is probably going to be the way that we go in the future. So these older factories are kind of at an advantage and a disadvantage because it's once it's built, you're invested, and it's very expensive and challenging to upgrade or change your lighting or hydroponic system at that point, so you're kind of stuck. But at the other hand, they've been doing it for a long time, and so they've really perfected their workflow kind of supply chain processing logistics there. So it is still a very young industry. Exciting discoveries are being made on a regular basis, and plant factories are starting to become economically feasible. As of last year, only 20% of Japanese plant factories were actually profiting, which is interesting. So a lot of room for improvement, which is a good thing in my opinion. So I came back from Japan and was ready to actually conduct an experiment in our own vertical farm facility, hoping to sort of implement some of what I've learned, some of the practices into our own facility as well. So the primary objective was to evaluate light intensity or daily light integral and the fertilizer type on biomass yield and quality of lettuce and basil in our commercial scale facility, with a secondary objective of gaining experience with crop production and labor management, sort of managing and working with my fellow students, gaining this operational experience in a commercial scale facility. So quick overview of our experimental design. So we have our 2 chambers here with 2 modules per chamber. One chamber was designated inorganic and the other organic. And then one module in each chamber was for lettuce and the other for basil. And then we have 3 different light intensities, starting with the lowest intensity on the bottom, working up to the highest on the top. We used spectroradiometer to ensure that our photosynthetically active radiation values were correct. We went with 200, 250, and 300 micromoles per meter squared per second, which is a daily light integral of 10.5, 14.4, and 17.1 moles per day, respectively. So that was kind of our setup. We wanted to see how all of these variables interacted. The lettuce was grown for 14 days in the seedling phase and then 28 days from transplant to harvest. There's an industry standard target weight of 150 grams that we sort of used for our target. And so my data here, the orange is inorganic and the green is organic. We have wet and dry weight of the lettuce. So the middle orange bar here actually is over 150 grams. So we reached our target weight at our medium DLI. We were able to hit that just at the middle level. The organic was lower, but we found out from the company that it was actually not a complete fertilizer and was quite low in calcium. So we did see that tip burn and we did see that reduced shoot yield. And we only kind of found that out after the fact, that it did not contain calcium there. So moving on to the basil, we gave it a few extra days. We had 16 days in the seedling phase for the basil and 35 days from transplant to harvest. So it's kind of like two-factor experiments being run in parallel. So our basil shoot weight again and the dry weight, just to compare, you can see actually that the shoot weight decreased as we went from 250 to 300 micromoles which is quite an interesting effect there. So because one of our sort of hypotheses was that more light might not necessarily be better, and perhaps there's a sweet spot where we find a balance between energy cost and productivity, because ultimately we're trying to maximize our profit, not our yield. So just another way to present this data, comparing it to area. So we have here grams per meter squared for both just the growing area and also the chamber area itself. And what we found is that the inorganic lettuce actually was producing over close to 8,000 grams per meter squared at our 2 higher light intensities, and that for all 4 of our modules, the grams per meter squared actually increased with our light intensity, which may or may not come as a surprise. So another look at this data, if we want to compare our fresh weight to our energy use, this data is in grams per kilowatt hour used for the transplant to harvest phase of 28 days for lettuce and actually 35 days for the basil. I have them together here. Lettuce is on the top, basil's at the bottom. So we can see that with the increased light intensity, we also have an increased energy cost that goes with that. So you can kind of visually see that it jumps up a little bit more from 200 to 250 than from 250 to 300. And I ran some statistical analysis on this as well, which I'll discuss. So basically, if you see our numbers for lettuce are around 40 to 50 grams per kilowatt hour used, we can compare this to Dr. Nadia Saibai, who's actually another SEAC alumnus who does engineering, specifically with heating and cooling systems and energy cost. She has a number from her presentation that she gave at the SEAC seminar earlier, basically of a number of 23.25 grams per kilowatt hour, where we actually are in the same range there, where it's actually a little higher on our number. Wageningen University in the Netherlands actually just put out another piece of information recently where the number is actually as high as about 100 grams per kilowatt hour. So we are sort of in this range, in this sort of expected range of energy use efficiency. And so that's very interesting to us for an economic standpoint. So I did run an ANOVA analysis on my analysis of variance on the weight crop yield, and it showed that it is absolutely significant. Light intensity and fertilizer type do both play a significant role per 2-factor ANOVA. So these factors do affect plant growth. And just to quickly conclude, again, back to the big picture. First, we developed a powerful distributed signal monitoring, processing, and control system for the UAG farm, enabling multi-factor experiments in this facility at commercial scale, and training students as well to be a skilled workforce. Second thing I did, of course, was receive hands-on training and skills for the daily tasks in the plant factory and best practices, as well as logistics and workflow, and actually the team culture, which is very actually strong in Japan, a very strong team-based culture, so nobody wants to be the weak link over there, which I think is an excellent perspective to view this through. And then finally, we carried out the very first research experiment in a commercial scale university research facility in North America, producing data on resource use efficiency and crop yield in the vertical farm, while gaining management experience working with graduate and undergraduate students. So it was a lot of fun, and while I was at the university, I had a few other achievements as well that I'd like to share. On the way to Japan, I stopped in Hawaii on the island of Hawaii, met with HI-SEAS, which is the Hawaii Space Exploration Analog and Simulation Program, initiating negotiations for getting them a lunar greenhouse module for their Mars habitat. After going to Japan, I went over to South Korea and spent a week with Seoul National University's College of Ag and Life Sciences with Dr. In-Bok Lee's lab, and that's an image there of his greenhouse research facility that's packed full of sensors, doing lots of interesting work on fluid dynamics and that type of thing. I also attended the Agri-World Tokyo Expo, which was an enormous trade show for all things agriculture. They had a whole plant factory section. You can see here full-scale vertical farming systems in a convention hall, and many suited convention goers. I also served as president of the Controlled Environment Agriculture Student Association from spring 2016 to fall 2017. Recruited lots of club members and sort of breathed some fresh air back into what was a non-functioning club at the time. We did many tours and have performed outreach on campus, inspiring, recruiting, motivating, educating students in both our program and interdisciplinary programs across campus. I also was a team member for the SEAC, providing tours and workshops for the SEAC short course, Summer Engineering Academy, other universities such as Chipingo University, and high schools in the area, doing more of this kind of outreach and education work. And also Dr. Kachira was kind enough to arrange a visit to Tsukuba, Japan, where he did research on aerodynamics, and I got to visit this governmental research facility, NARO facility, and was really exposed to some higher level agricultural engineering projects there. So just some acknowledgements. I had a lot of help doing this. I would like to thank my committee members for serving on my committee. I really appreciate it. Many other professors, both at the SEAC and at Chiba University, also played a major role in my accomplishments. Of course, Miles Lewis of Arizona Vegetable Company was a priceless consultant for us, really helped us out a lot. And then my fellow students, Brian Kaplan, was there on a daily basis with me managing the facility, and basically the rest of Dr. Kachira's lab and some club members from the Controlled Environment Student Association as well. And some industry folks, Paul Harday for his priceless contributions to getting our facility up and running. My family, of course. So I do appreciate it, and thank you for listening, and I will be more than happy to answer any questions that you might have. Go into more detail on any part of it.
Nice job on the timing, 35 minutes. Great. So that gives us a chance to have maybe a couple of questions from our audience. Rafi, may I give you the microphone so we can also hear?
Alex, I want to congratulate you first of all for a terrific presentation. I've kind of followed you from the sidelines over the past 2 or 3 years, and I've seen tremendous, wonderful development of your knowledge base and your career, your presentation style, and both you and your mentors should be really proud of the work that you have done together. Now, I have a question that links what you said about the facility in Japan, which basically you described as being almost sterile, leaving aside the growth of algae. And specifically, you mentioned that the facility was free of insects, but you said nothing about whether or not there was any kind of microbial populations in the root area. And I'd like for you to comment on that and see if you can then link it to the controversy surrounding the designation of hydroponic crops as being organic versus non-organic according to the USDA, based primarily on the presence of microorganisms in soil. So, your impressions.
Yeah, thank you, Rafi. Great question. So, I did ask as many questions as I could about the management practices and sterility. To my knowledge, they don't add any sort of antibacterial or antifungals to the nutrient solution, but simply through their very strict sterility practices, including putting on rubber gloves once you get inside and even cleaning the gloves before you start to work. They're effective at not introducing those microbes into the solution in the first place. Now, obviously, the flip side of that is they're not using any beneficial organisms either. But it seems that their strategy is working. The roots are very clean and white and disease-free, and no pathogens present there. The only pathogen that I discovered was on maybe like one head out of 1,000. I would find a very small amount of powdery mildew, and they would actually sort of quarantine that and ensure that it doesn't spread. And again, with the algae, like you mentioned, that is obviously an organic organism growing in the water that's not desired. And any strategies that they might implement to control that would also control any beneficial organisms or live organic nutrient solution that they were using. And then relating that to the sort of current controversy with hydroponics and organic, and that is absolutely a huge issue right now. There's a lot of industry interest in using organic fertilizers in hydroponic, both for economic and sustainability reasons. And really all I think I can say about it is that in my opinion, the best avenue to take would be to develop both from like a legal angle and also a technical angle, a way that we can grow hydroponics using organic, biotic nutrient solutions in a way that improves our productivity and also enables us to get credit for what we do, which is, of course, label organic certified, and that actually creates an opportunity there to use waste biomass to actually turn into and create our own organic fertilizers. I hope that helps.
So on the third part of the third chapter, you were talking about resource use consumption. It looked like from 250 to 300, there wasn't really a lot of gain in terms of the lettuce. I think it gained about 20 grams or something. I was wondering if you were able to tie an economic cost to the production of each head at each light level. And then I guess kind of a follow-up question is, when you were talking about the kilowatt hour consumption, is this inclusive of HVAC and other utilities, or is this simply just the illumination?
Thank you. I should clarify. So for what I use to calculate the number of kilowatt hours used per gram of average head weight, I took into account the HVAC system, the lights, and the water pump and air pump.
Do you have an idea of what the production cost was for a head of lettuce in your production system?
You could get that information from the numbers I have. What I have here is in a per gram. So if you were to take that and put back in our average grams per head, we would have an energy cost per head.
Just may I add to that? The short answer in terms of the cost analysis is no, because there's no cost for labor or fertilizer or other kind of supplies other than the electrical, the cost for the electricity, which can be calculated as Alex said. But I think we need those kind of evaluation and numbers, values, and get it out there.
Please.
I was just having trouble hearing you. The mic didn't seem to project. Is this?
Yeah. So what I said, in terms of total cost for the production, the short answer is no, because I don't think we included the cost for labor or CO2 or other materials used, other than the cost for the electricity that we can calculate because we have the numbers, but we can guess pretty much based on what we used, but it was not included in Alex's study. But we need those types of information in vertical systems and get the information out there, which is really needed. Chance to others as well, Rafi. Yeah, if no, then
So, usually in these kinds of presentations, people always ask about the future. So I'm interested in two aspects of the future from your point of view. First, what do you think needs to be done to vertical farming in order to adapt it to growing plants, crops other than lettuce and microgreens? For example, tomatoes. And secondly, what are your own plans for the future?
Sure. So, to your first question, how can we grow more than just leafy greens, herbs, and microgreens in these facilities? I believe that over time, as the cost of lighting and lighting technology improves, and the cost of lighting decreases, and maybe alternative technologies start coming into play, such as using solar radiation, even in an indoor setting, through either fiber optic or more efficient photovoltaics, because crops like tomato have a much higher daily light integral requirement than our leafy greens do. It's just not cost effective yet to do so. But as our also hydroponic system technology and everything improves, we should be able to balance out the sort of cost and revenue to be profitable. I know that strawberries are starting to be grown indoors, which is promising. My own plans for the future actually are to return back to Japan in January. I have a position there that I'll be starting at the beginning of the year, so just in a few short weeks. Actually, Ray's Farm and Chiba University and JPFA are starting a new pilot program, which is a research project on plant phenotyping with artificial intelligence. So I'll be a sort of technician, research assistant type of role over there.
may I add one more thing to what you said? So Alex is going back to Chiba University. We are really happy for that, and I would like to mention that we have a long history for collaboration and interactions with Chiba University. I want to acknowledge and thanks to my colleague and friend Dr. Cherry Kubota. She maintained that interaction and collaboration officially, and we were recently asked to keep that program ongoing, and we respond to that yes. So kind of received the torch from Cherry. So if there are interests in terms of internships, being able to go there and then work just like Alex did, please come and have a discussion with us on that, so we can create those opportunities, not for internships. Also, we are looking, furthering our collaboration for research opportunities too. So we're going to maintain that, and you being there, I think, is going to be advantageous for us. So you can be our ambassador there to really keep that program going and grow it further.
Yeah, I guess, and to your point, Rafi, I would say as far as for future, I think that the more we can sort of develop a worldwide network and connect these different wings of the industry that might be operating fairly independently, that can only help facilitate like collaboration and innovation in the industry. So that's part of my mission going over there, to try and further bridge this gap.
Questions? Ryan, Neil, wonderful.
Your system was a closed system in the indoor farm, correct?
Yes.
How did the chemistry of the water change from the initial mixture to, say, your final? I'm assuming you didn't do any water changes between first day and last.
We did not do water changes, but we did do like replenishing the nutrient solution, and so we would add more fertilizer concentrate when we replenish the nutrient solution. So actually, as far as the nutrient balance profile, it stayed pretty uniform throughout the course because we did top up with nutrient solution. Is that your question?
I was wondering more if any elements were accumulating since they're not being taken up as much as other elements. You don't see anything like that, accumulation of sulfur, perhaps, sodium.
Nothing so drastic to like draw our attention.
All right.
Last chance.
Good. Anybody? Last chance? All right. Well, okay then. Thank you very much. And thank you, Alex, for sharing your experiences with us and with our audience here. And thank you very much again for being here. Please enjoy the food and goodies and drinks back there. So we're going to take about 5 minutes break. Then we'll have our committee kind of spending a little bit more time with Alex. Thank you so much.
Thank you, guys.
I would like to acknowledge and thank Austin for helping us to make these arrangements all the time. He's here to help.